Nerve conduction. So it is a topic which describes about how the messages are passed in our brain. So nerve conduction is nothing but passing of nerve impulses along nerve fibers. It is called nerve conduction. So the process of passing nerve impulses through the neurons is called nerve conductions or neuroconductors, conducting can say. It involves the brief electrical fluctuations that propagates down the neurons, dendrites, then through the cell body and finally to the tip of its axon. Nerve cells generate electrical signals that transmit information. Though the nerve cells are not good conductors of electricity, they have evolved elaborating the mechanisms for generating electrical signals based on the flow of ions across their cell or plasma membranes. So like that there will be passing of these ions, sodium, chloride, calcium or potassium, etc. Because this is the way. So the sensation and perception depends on conduction, the coordination of the both sensory and motor functions. So commonly what sensory organs will be bringing the messages from the outworld to brain, and again they are interpreted and again they are carried, and in the form of muscular action the activity is done. This is a way of efferent or efferent also we can say, bringing the information to the brain and taking the information from the brain to the muscular organs to do some activity. So the coordination of both the sensory motor functions of the body and thus exchange of information takes place at the level of nerve conduction in our brain. Coming to synapses. What is synapses? It is a place exactly where the exchange of information is taking place in our brain. Synapses is a place where information is passed from neurons to nerve or neuron to neuron. So it's the junction where the information message is shared and exchanged. It is a structure which permits a neuron to pass an electrical or chemical signals to one another and or targeted effector cell. Synapses is converting the electrical signal into chemical signals in the form of neurotransmitters release. Okay, like example acetylcholine, that is one neurotransmitters, or we can say dopamines. They will be doing main actions in our brain, and again backing the signal in the form of electrical form. So this is something what is happening at the level of synapses. If you see this diagrammatic representation, this is called a nerve cell, and this like tails will be having a dendrite, small one. The lengthy one is called as axon. Then it is another nerve cell. Then how the information is taking place? Okay, from dendrites to dendrite, that is called dendritic synapse. From one dendrite of one nerve cell to dendrite of another cell, the information is passing. That is called, that junction is called that dendritic synapse. When the information is carried from one directly nerve cell to other, that is called neurotransmitters are released at that point. Then coming to the types of synapses, excitatory ions channel synapses, and these synapses have neurotransmitters that are sodium channels. Then inhibitory channels responses are chlorine channels. Non-channel synapses, then neuromuscular junctions, electrical synapses. It is the protein channel called gap junction which directly connects to neurons, and fast conduction and biodirectional and transmission of electrical impulses and molecules. The stages of nerve conduction can be rising potential, threshold, rising phase, failing phase, recovery phase. By using all above stages, signals arise at cell body, across the synapses or through the dendrites, stimulating neurons to generate its own signals sent along its long axon to other neuron cells or muscle cells. The nerve conduction study measures how fast electrical impulses move from one nerve to other nerve. It also can decide if any nerve cell damage is seen in the brain. So this is about a few points related to nerve conduction.